Well, first I'd like to express my joy and delight and overwhelming sense of humility at being honoured with this award, to follow in the footsteps of my dear friends Tuck and George and many others is an absolute delight. It's been a joy to be associated and to have attended numerous meetings sponsored by the Foundation IPSEN, and I'd like to express my warm thanks and appreciation to Eve and Jacqueline and to the Foundation itself for the exceptional role that it plays in the world of science. Now, what I want to do in this talk is to have fun. So I'm going to give a talk that will cover science, but I also want to be a little bit provocative because I remember when I first met George Martin at the outstanding Gordon Conference on Aging that he organised back in the late 1970s. I think George described me subsequently as a very outspoken young man. And I guess what I want to do is to show you that I can now be an outspoken older man, and to try to bring some challenges to our collective effort to understand this most remarkable process, which is aging and its biology. So enough of the introduction. I want to begin at the beginning, and I think in a moment you'll recognize what you're looking at here. It's not a picture of the sun. This is a wonderful photograph from the Wellcome Trust collection of an egg at the moment of fertilization. This is the point at which life begins. But actually, although it's the point at which our lives begin, it's not the point at which life begins, because the cells that we can see participating in this glorious fusion are cells that came from the adults that have participated in creating a new life, that came from cells within the bodies of their parents, from their parents and their parents and their parents and their parents. So what we are witnessing here is the continuation of something which is quite remarkable and which is represented by each and every one of us in the room, and that is the continuity of cellular life on this planet. If the records existed, each cell in our body could trace its ancestry through an unbroken sequence of cell divisions that goes back billions of years to the emergence of the earliest cellular life forms on this planet. So in that sense, each of us is truly amazing, and this phenomenon that we see here in front of us is absolutely astonishing. Of course, there is an old joke about life being a sexually transmitted condition with an invariably fatal outcome. And to some extent, it's the invariably fatal outcome that we want to talk about. This is, of course, the process of aging. There has long been a tendency to sort of link reproduction and death. It's worth just reflecting that that miserable old philosopher, Aristotle, left us with the idea that each sex act has a direct life-shortening effect. When you look at him here, he looks as if butter wouldn't melt in his mouth. But he sort of introduced this idea that has lingered through the centuries since his time. John Donne, one of the greatest of the metaphysical poets, writing in the early 17th century, after a life of considerable sexual activity, one might even say excess, became a priest. And he wrote a poem that included these wonderful lines that are very hard to read, but I'll try. Since each such act, they say, diminisheth the length of life a day, I'll no more dote and run to pursue things which hath damaged me. And then, come into the 20th century, and we find Sigmund Freud. We're very familiar with Freud's enunciation of the life principle, eros, but Freud was also responsible for writing about the death principle, the death instinct that he called thanatos. And Freud was explicitly referring back to these kind of ideas that originated with Aristotle and had been kept alive and are still alive today. There are quite a number of people who are familiar with the concept of the sexual orgasm as the little death. This idea comes from Aristotle. Now, I don't know whether there are any fruit flies in the room. If there are fruit flies, then the sad truth is that for fruit flies, this means something. But for the rest of us, we can forget Aristotle, John Donne, and Sigmund Freud, and we can party, because we know that sexual activity does not shorten life. But I'll now move on to the more serious side of stuff. I'm thinking about the continuity of cellular life, and this was really the point at which I began to grapple with the scientific basis of aging. I started at the prompting of Robin Holliday, whom I'd met the year before, looking at a key question, and that is how a cell manages to maintain its molecular integrity from division to division to division. Now, that led to some quite detailed modeling of how the machinery of the cell might give rise to mistakes in the molecules that it synthesized. And so a key question was the extent to which the cell could either amplify the mistakes, which could occur through processes of positive feedback, or sort of quench those mistakes by investing in molecular proofreading and correction. And emerging from this was a curve that looks like the one shown here, where what we're plotting is the molecular integrity of the next generation of cells plotted against the molecular integrity of the parental generation of cells. The line that bisects this diagram is the line where the molecular integrity would be the same from one generation to the next. And the key thing is that if you are a cell which starts out with a high level of molecular integrity, what I was able to show in this model was that you could reach a point that would be stable. That's the upper right of the little black circles. And if you were at that point, you could manage to sustain a certain level of damage, a certain level of error in the macromolecules, but it need not increase. If some perturbation, some accident, took you down towards the lower of the black dots, then you would be in trouble, because at that point you would be deteriorating in the molecular integrity from generation to generation, and things would fall apart very quickly. So this was an interesting way at looking at the sort of stability and instability that existed within cellular life. The margin of safety this modeling showed, which is the difference between these 2 points, would be increased by investing more cellular energy in molecular proofreading and error elimination. But on the other hand, if you were to decrease that investment in molecular integrity, then that margin would be reduced. Now, if you reduce the margin, which effectively is pushing the curve sort of downwards and to the right, you could reduce the margin of safety, reducing it to a minimal level, would save a very considerable amount of energy within the cell, because you have a strict law of diminishing return. You have to put a lot of energy in to make the cell safer. So it would save a lot of energy, but it would leave the cell highly vulnerable to the accumulation of deficits. So this was kind of an intellectual starting point. And at this point, I was really just interested in the kinetics, the dynamics of this process. But then I became aware of the writings of this extraordinary biologist, the German naturalist August Weismann. And Weismann left us with a most important insight into biological systems, and that is the difference in a multicellular organism such as ourselves between the cells that constitute that immortal germ-line that I referred to at the beginning and the rest of the cells of the body that make up what Weismann called the Soma. And Weismann had the ability to see that the heart of the puzzle of aging centered on what distinguished the immortal germ-line from the mortal Soma. And that provided a trigger that was particularly exciting. From that, and I'm going to summarize the outcome of the thinking about this, I was imagining what would happen to the earliest cellular life forms. They would be pretty inaccurate, and one of the challenges that life faced in those early years was how to lift themselves up by the bootstraps, how to evolve better repair. And we know that happened. We know that cells became more sophisticated in error correction in proofreading, and this would have been a process that would have been driven very strongly by selection. And then again, going back to the sort of the dawn of life on the planet, we would have seen the emergence of important distinctions. For example, some organisms remained unicellular while some became multicellular. And we have the descendants of the unicellular organisms and of the undifferentiated multicellular organisms still with us today. But something very interesting happened in the ancestors of ourselves and other species, and that was that there came a division of labor, that only a subset of cells within the body were charged with the reproductive activities, while the rest were left free to become specialists, to become the neurons, to become the heart cells, to become the kidney cells, become the liver cells, become the skin cells. And what I suggested in this paper back in Nature in 1977 was that for the kind of energy-saving reasons that I've just alluded to, for the somatic cells that were not going to contribute directly to the next generation, you didn't need to invest so heavily in accuracy because it was not important to maintain the capacity to propagate indefinitely. And this was really, sort of the beginnings of this journey. Now, there are exceptions. Biology is wonderful. We heard yesterday in Steve Austad's glorious lecture about the power of comparative studies. Steve was talking mainly about higher organisms, but if we go down to the sort of zoo of simple animals, the invertebrates, we discover there is an incredible richness of species that live their lives in many, many different ways. And one organism that exhibits a very different way of living life is the hydra. The hydra, when it was first characterized, caused a great debate in the 18th century because nobody could tell whether it was an animal or a plant. If you take a hydra and you cut it up into small pieces, each piece regenerates a whole. So there was a very vitriolic and divisive feud within the life sciences about whether hydra were animals and plants. And a Belgian scientist, Abraham Tremblay, spent a lot of time being brutal to hydra, cutting them up into small pieces and abusing them in all kinds of ways and demonstrating that each piece had the ability to regenerate a whole. More recently, Daniel Martinez has shown that if you follow hydra across the years, you see no evidence of intrinsic aging. Now, the interesting thing about hydra is that they do reproduce by budding. Any part can regenerate the whole. Germ cells permeate the structure. Therefore, the hydra has no true soma to be disposable. So this is an interesting exception that, as it were, proves the rule. In that original Nature paper, I also speculated really about the distinction between Germ-Line and Soma, and these are some words that come from the end of the paper, that accuracy in the Germ-Line is vital, but a high level of accuracy in somatic cells may be a luxury our genes do better to forego, and proposed that aging may therefore be the result of switching off the mechanisms responsible for high accuracy at or around the time of differentiation of somatic cells from the germline. Now, over the years that have accumulated since then, we've learned quite a lot about how germ cells do in fact maintain their molecular integrity to a higher degree. We know about telomerase, for which the Nobel Prize was awarded 2 years ago, not 3 years ago, recently. We know that telomerase is active in germ cells and in some stem cells, but inactive in most somatic cells. We also know that embryonic stem cells show lower mutation rates than embryonic somatic cells. And it was particularly delightful a few years ago when, in a sense quite independently of the prediction that I had made, my colleague Gabi Saretsky in Newcastle was studying mouse embryonic stem cells. You can see the embryonic stem cells at the top left, and then you can see when these cells are made to differentiate and become embryoid bodies, day 2, day 4, day 6. What Gabi discovered was that there is a comprehensive tuning down of the complete array of cell maintenance systems, of the antioxidant defenses of DNA repair of chaperones, and you can see that markers of oxidative stress, for example, cellular peroxides, increase dramatically over the first few days of differentiation. Then 4 years later, she showed the same thing in human embryonic stem cells. So it's not often in science you get the chance to say, I told you so. But this was one of those delightful occasions. I think Sometimes when we focus on aging, we forget that actually the bigger puzzle is to explain how it is that the germline truly sustains its immortality. And I think looking at this is important because it helps us get insight into what it is that aging is really all about. So I'm not going to go into this area briefly, but I think we can understand broadly the three ways in which the germline preserves its immortality. The first I've touched on. There is greater investment in maintenance in germ cells. They are important. They are looked after more carefully. Second, there is stringent quality control. We know that in the male germline, there is massive overproduction of sperm. And we can be pretty sure that when the race is over and the one sperm has fertilized the egg, that it will be in good physical condition. It wouldn't have won the race otherwise. The egg plays a more passive role in the act of fertilization, but there is quality control going on all the time in the ovary. At the time when the ovary completes its development, which happens very early in life, there are many millions of potential oocytes. But in the case of a woman, when around the age of 50 the menopause occurs, there are really none of them left. Now, the consumption of oocytes through ovulation accounts for a tiny, tiny fraction of the number that have gone. So where have the rest gone? Well, we don't know the details, but it appears that there are active mechanisms for quality control. When oocyte activation occurs, quite a number of oocytes respond to the signal to start to mature. But only one of them, or two if you have twins, three if you have triplets, actually goes through successful ovulation. The rest are deleted. We don't know the criteria by which the deletion occurs, but there are very suggestive pointers to the role of the mitochondrial bottleneck and strict screening in terms of mitochondrial quality. So quality control is very, very important, the kind of thing that you cannot afford with somatic cells of the body. And then something else is very important, that is the selective elimination of defects. Now we know that selective death occurs at all stages through the reproductive cycle, and of course selective death occurs on the rare mistakes that make it through to a new individual. Some of these attributes are shared by adult stem cells, and I think one of the really interesting areas when we look at potential for regenerative medicine is to understand, so how effective these mechanisms may be, and to recognize that stem cells, like germ cells, are vulnerable to the accumulation of damage. So we should not be too complacent. For example, very exciting work published recently on the generation of IPS cells, induced pluripotent stem cells, induced grown in culture. What we don't know is what genetic damage those cells may carry that could have an impact on their effectiveness to regenerate organs and tissues if they were to be used therapeutically. There's a great deal we need to understand, and this interface between germline immortality and somatic cell mortality is going to be one that we need to understand much, much better. Now, lifting ourselves up and looking at the organism from a rather more sort of elevated view, another essential issue that we need to consider when we think about how organisms respond to the pressure of natural selection is to recognize that what Darwin taught us is that if we think of the organism as a black box, it's a black box that takes resources from its environment, primarily in the form of nutrients, and it's got to deliver an output of progeny, and the law of natural selection, evolution through natural selection, tells us those organisms that are the most effective in transforming nutrients into viable progeny are the ones that are going to be most numerous in the next generation. But in doing this, organisms have to make tough choices. So let's take the lid off the black box, and we can see that organisms have to allocate resources to growth, to maintenance, repair, to storage, to reproduction. And the fundamental principle is that it doesn't matter how much energy you have, you still face the issue of how you optimally allocate the energy between these different activities. And the disposable Soma theory can be restated as saying that the optimum level that you invest in somatic maintenance and repair will be sufficient to carry the individual body in good shape through the kind of lifespan that it can expect in the wild, where there are many sources of death that are effectively inescapable, so lifespan is curtailed by that, but they will not be sufficient to provide the body with the capacity to last indefinitely. So what we predict is that the period of longevity assured by the investments that we make in maintenance and repair should be tuned to the ecological circumstances of the species in question. And we can see how this relates to the evolution of longevity as we see it in different species. Because if we conduct a thought experiment, and we imagine that we have some hypothetical species, perhaps it's a form of mouse, has high levels of extrinsic mortality, so it dies away quickly in the natural environment. Because of that, it's not worth investing massively in cellular maintenance and repair, and as a consequence of that, if you take it to a protected environment, it has a relatively short lifespan. Now we suppose that that species evolves an adaptation. Perhaps your little mouse becomes a little bat. As a little bat, it can escape many of the hazards of the environment. It can, for example, forage over a greater range. It will have better survival across the age range. But now we can see that if nothing changes, your new bat has a problem, because it has the potential to survive. But now significant numbers of individuals within this population are going to fall apart because their maintenance is not sufficient to keep the body in good shape through the extended expected period of survival in the wild. So what does the bat need to do? It needs to increase the investment in maintenance and repair, and as a result of that, it increases its intrinsic survival capacity. So in this way, and this does relate to what Steve Astad was talking about yesterday, we can begin to get to grips with understanding the evolutionary divergence of lifespans within species. Some years ago, Pankaj Kapahi, now working at the Buck Institute, when he was a PhD student with me, he produced these lovely data. These were taking cells from species of different lifespans, mammalian species, and then throwing bad things at them. And what the data showed was that the cells from the longer-lived species were better able to withstand the bad things. They survived better. There was a strong positive correlation between ability to withstand stress and longevity. So the mechanistic implications of this concept, the disposable Soma theory, reach quite a long way. There's a difference in maintenance and repair between Germ-Line and Soma. Aging is caused primarily by damage. Longevity is regulated by resistance repair. It also predicts that the aging process is inherently stochastic, that there will be multiple mechanisms and therefore complexity, and it also has some interesting things to say about suggesting some plasticity and trade-offs. So we can carry on from this to sort of make some inference about the likely genetic control of longevity, and the consequence is that we would expect longevity to be regulated by the actions of natural selection, working on multiple systems for molecular maintenance and repair, and harmonizing more or less how good these are in terms of the kind of lifetime that they can sustain. And this will apply to the whole spectrum. There will be hundreds of genes that will be involved with this. And very excitingly, what we've learned from the recent work, particularly with nematode worms, fruit flies, and a little bit with mice, is that organisms also have evolved some plasticity to respond to what the environment offers. If the environment is bad, as for example is the case when we see nematode worms switch themselves to the alternative developmental pathway that produces the Dawa larva, the long-lived, enduring, stress-resistant dispersal form, the organism is making a switch from investing in growth and reproduction to investing more in maintenance and repair for the time being. So there is plasticity that is regulated in some species by a kind of evolved genetic architecture for doing this. So this again is exciting. So what we see is that this allocation that I showed you earlier is modulated by environment and other factors. And we know that the much studied insulin signaling pathway, nutrient sensing, has important effects. Insulin IGF-1 signaling promotes growth and storage, and low insulin IGF-1 promotes cell maintenance. So these can be related directly to these allocation issues. We see this comes from, it's now a 5-year-old review, but the ideas are still basically valid, from Christensen and colleagues, showing on the left-hand side the players that are involved in pathways that regulate nutrient sensing and metabolism in worms, and the homologs that exist in humans. Now, a lot of attention has been focused on these, but actually the focus is not really on aging. The focus is on longevity, and that's very important, because the aging process happens just the same, just later. It doesn't actually alter aging. The aging phenotype is at the bottom. And this, I love this diagram, because right at the bottom is a box that says many downstream targets. And this is really like the old explorers' maps of the world, which showed the known world marked in some detail, and then very speculative contours of unknown continents with sea monsters and everything populating the map. So many downstream targets is actually where it's at in terms of the mechanisms that drive aging. Another very important thing that comes out of the observations on nematode worms relates to the agenda that Tuck mentioned in his kind introduction, and that is the important role of stochastic variation. That's non-genetic, non-environmental variation. Now, this is a figure that we redrew using data from Tom Johnson of a population of wild-type worms in the black bars and age 1 mutants with extended lifespan in the bars on the right-hand side. And what we can see is that, for sure, there is increased lifespan associated with the age 1 mutation. No question about it. It's really interesting that you see this increase in lifespan. But when you plot the data this way, what you see dramatically is the huge individual-to-individual variation in lifespan that can't be explained by genetic variation within the populations. These are isogenic populations. Within each population, the individuals are genetically the same. It's very hard to explain in terms of environmental variation, because you can make the environment as uniform as you like. You get the same results if you perform the experiments with stirred liquid cultures. So the variation is intrinsic to the biology of the organism, and we still don't understand it. There are the beginnings of some results that explain this variation. You know, we need urgently to get a better handle on what explains this underlying variation. And this brings us into what I call the deep mechanisms of aging. And these are the mechanisms that relate to molecular integrity, that relate to continuing to sustain the processes at the core of life: DNA, replication, transcription, translation. We know that each of these processes is vulnerable to errors. We know that some of those errors come from intrinsic mistakes in synthesis, some come from damage from reactive oxygen species. We know also that because these sources of damage have been around since the beginnings of life, that our cells are equipped with potent defenses against these systems. There is a cyclicity about this, and this is really where I started thinking about how the molecular integrity of one generation depends on the molecular integrity of the preceding generation, initially just in the context of protein synthesis, but this idea can be generalized to include molecular integrity of all species within the cell, molecular species. And there is also the important role of the mitochondria. You know, relatively early in the evolution of our ancestors, a remarkable event occurred when bacteria became endosymbionts within the cell, and these leave as descendants our mitochondria. The mitochondria are terribly important because they are the primary source of intracellular energy, but also they're the primary source of intracellular reactive oxygen species. And I think there are deep questions. You may have seen earlier this year in PNAS, Axel Kowalt and I developed a hypothesis that I think gets to the heart of what is going on with mitochondrial damage accumulation, and it explains possibly, if it turns out to be correct, the significance of the rapid fusion-fission cycling that occurs within mitochondria. But that's too complicated a story for me to sort of digress into now. Now, we see when we look at cells and their aging, this remarkable phenomenon that was discussed yesterday in recognition of this being the 50th anniversary of Len Hayflick's discovery of the phenomenon of replicative senescence, that cells, when grown, can divide and divide and divide. They then seem to stop, and beyond that point, they don't just die, they survive for quite a considerable period, during which other changes may occur. And George Martin was one of the pioneers in really understanding the complexity of what is going on here. I think there are many concepts that come from some of that early work that we need to revisit. George particularly gave us the concept of how clones kind of grow but attenuate. And I think understanding the heterogeneity within cell populations is something that we're coming back to. 30 odd years ago, Smith and Whitney published a remarkable paper in Science in which they showed that if you take a single fibroblast and you grow it into a large population, and you then culture it, and at different points in the lifetime of that culture, you take a sample of cells, so early, middle, late in the culture, you pick 200 cells at random out of the mass population, and you grow those individual cells, then you get the data shown on the left-hand side of this diagram. What you see is that although the cells are the direct descendants of a single founding cell, they exhibit extraordinary heterogeneity in individual division potential, with quite a strong indication, certainly at the early and middle stages, of bimodality in the distribution. Now, this is a challenge to any simple explanation of how these cells age in terms of, the regular shortening of telomeres or anything like that. So 10 years ago, with a mathematician, Peter Sozo, we developed a model based on what we knew of the biology of these different mechanisms, built a model that incorporated different mechanisms running in parallel, synergizing and interacting. This was oxidative stress, mitochondrial DNA mutation, mutation in the nuclear, in the non-telomeric parts of the chromosome, and the erosion of telomeres. And what we found was that with the model that incorporated these multiple interacting partners, and only with a model that incorporated multiple interacting partners, could we reproduce the heterogeneity that had been reported by Smith and Whitney. So this was kind of provocative, and a few years after that, João Passos joined the lab. He had been looking for a challenging PhD project and came with a sort of gold-plated government scholarship from Portugal. And I said, right, if you want to do something challenging, why don't you test the idea that mitochondrial mutations might be accumulating in cells and be contributing to some of this heterogeneity? Because this is what the models had suggested. And as many of you will know, João produced a remarkable body of work which demonstrated that, yes, indeed, there is massive accumulation of mitochondrial dysfunction with replicative senescence, and it accounts beautifully for that stochastic heterogeneity in telomere-dependent senescence. But João was one of those bright people who is never content to leave well alone. So João went on to do other experiments, which involved, in the first place, messing with the telomeres and showing that then the mitochondria went bad, irradiating cells, damaging the DNA in cells, and showing that the mitochondria went bad. And this established that we have these complex intercommunicating cycles of damage that led to the conclusion that Tuck alluded to in the introduction, that actually cells are equipped with a rather remarkable capacity to sense and integrate signals from multiple different kinds of damage. I guess with hindsight, it's not surprising that it's so. I mean, science is brilliant, but we spend our time blundering into discovering what should have been obvious if we'd only thought about it right in the first place. And this is one of those cases. So what we found is that cells have this amazing capacity to pick up on damage that may start in the mitochondria in one cell. And in the cell next to it, they may start from the telomeres becoming critically short. And in the cell beyond that one, they may start from having DNA lesions on the non-telomeric parts of the chromosome. Integrates the signal. And this tells the cell that it is in a wobbly state where, so just as in some cells, those kinds of signals are read as demanding that the cell be destroyed completely, producing an apoptotic response. With the majority of cells, what you get instead is a different response. You get replicative senescence, where everything gets kind of locked down. So I put in my title, what do we really know and what do we really want? And I'm coming to the point now where I'm going to stop saying what we know and I'm going to move beyond the kinds of things that sort of are established from this starting point that Tuck alluded to. What we really know, we know a few things. We know aging is not programmed. We know aging is driven by multiple types of damage. We know aging is seriously stochastic. And we know aging embodies trade-offs. Of course, we know quite a lot else, but those are sort of broad categories. And I want to ask now about things that we don't know well enough. Do we really know what is driving the ongoing increase in longevity in human populations? These data, this comes from the paper by Open and Verpel in Science in 2002, show how the increase in life expectancy, which was forecast by all the best demographic forecasters in the world to be about to hit an imminent ceiling back around 1980. The reason for that was that up until that point, one thing, and really only one thing, had been recognized as the driver of increasing human longevity, and that was the declining early and midlife mortality. But something totally new showed up. We have no further gains to make by reducing early and midlife mortality in the developed countries of the world. If you could eliminate early and midlife mortality, you would see a gain of only a month or two in life expectancy because so few deaths occur before advanced age. What is driving the increase in life expectancy now is declining the later life mortality. And we have some ideas about what may be causing it, but we really do not know. What we do know is that the aging process, if we understand it in terms of the lifelong accumulation of random molecular damage, the build-up of cellular defects resulting in age-related frailty, disability, and disease, is something which is actually much more malleable than we used to believe. It can be acted on by lifestyle factors. It can be acted on by environmental factors. We know that stress, environment, poor nutrition, they don't just sort of make you a little bit unhealthy. It seems that they drive the accumulation of damage which underpins the aging process, at least to some extent. But we know that healthy lifestyle choices, exercise, good nutrition, is important. We are learning about the importance of inflammatory reactions. Finch and others have told us a great deal in recent years about the importance of inflammation and anti-inflammatory factors that can quench those sort of fires of inflammation that cause damage. We know that socioeconomic differences make an enormous change to our lives. This, I must acknowledge my colleague Carol Jagger, who's in the audience here, for help in preparing this. Newcastle has a metro system. It's not the same as the Paris metro, it's not the same as the London Underground, it's not the same as the subway in New York. But it is similar in the sense that if you get on the station in the posh part of town, Pontyland, and you ride the metro for a few stops, you shed years of life expectancy and healthy life expectancy. So go from Pontyland at the top left to Byker, the bottom right, and you lose more than 10 years, 11 years in fact, in the age of expected onset of a limiting long-term condition for a 55-year-old. These are some of the most important issues that we face within our field, is really to understand what are the root causes of these differences in life expectancy and healthy life expectancy within our population. We could increase life expectancy in the US, in the UK, and probably in many other countries if we could simply bring the disadvantaged folk up to the levels of health and life expectancy enjoyed by those of us who have more of the good things. What exactly are the links between aging and disease? Now, we're beginning to see exciting progress, and some of it's been reported in this meeting here, about going back from the end-stage pathology that tends to dominate our attention when we look at a person with age-related disease, to beginning to work upstream in terms of the causal pathways. We've heard, again at this meeting, interesting evidence presented about the possibility that, for example, in the case of Alzheimer's disease, the amyloid plaques that we see are not the problem. They're actually a sign of a part of the solution The problem may be elsewhere in what's going on within the cells. So it's really important that we understand what are the upstream factors, because that's where the likelihood of, the effectiveness of interventions is going to be greater. And I think the excitement is that as we work up into the headwaters of one particular age-related disease, we find that we are in the headwaters of maybe several others. And this is tremendously promising, because this is where interventions may have their biggest impact. In Newcastle, we've been running now for some years the Newcastle 85+ study. This was started in 2006 when we approached everyone in Newcastle and North Tyneside who was born in 1921. It's quite a business to get the ethical approval to do this, but for those of you from the United States, there are great advantages to having a national health service and socialized medicine, because once you get the permission, you just simply go to a computer and you get a list of the names and addresses and general practitioners of everyone born in 1921. So we approached them. We got a very high proportion to agree to take part. We discovered in some pilot work that if we had asked them to come to a clinic to be assessed, only half of them would have done so. So our superb research nurse team went out to do all of the assessments in their homes. These were very comprehensive assessments. They included things like echocardiograms. They included a whole battery of tests and measurements that were made. We're just beginning to get to grips with analysing the data. But one important finding comes out of this, and that is that out of the 1,041 individuals included in the study, we looked to see whether they had or did not have each of 18 different diseases, and we found that no one at age 85 had perfect health. There was no one with a disease count of 0. Most people had 4, 5, 6 diseases. Nevertheless, well, the important comment here is that normal aging is a state characterized by extensive comorbidity, and I think one of the challenges that we face in doing research in this area is how to meaningfully investigate comorbidities and investigate treatments that will work where comorbidity exists. Nevertheless, and this was also quite a surprise, when we asked people to self-rate their health, we found that 4 out of every 5 of the respondents rated their health as good, very good, or excellent. So this was an opportunity to look at the population as a whole, not just to focus on the attention we had from hospital records of people who had come to our notice because they were sick. We had last year a lunch to which we invited all of the surviving participants, and I've never spent a more enjoyable day than in a room with several hundred extremely dynamic, extremely curious, extremely demanding. They were then 89-year-olds. These are some of them. How to measure the progress of aging. I think we know that we really want to be able to come up with effective biomarkers that can measure the course of aging. Alex Comfort, who many of you will know is a distinguished gerontologist, as well as being the author of The Joy of Sex, wrote in a very far ahead of his days article in The Lancet in 1969 about the importance of developing a test battery to measure human aging rate. And we know that here the National Institute on Aging has invested a lot of effort in developing biomarkers. People are trying to do this, and it remains an elusive goal. Within the Newcastle 85+ study, we tested 74 biomarkers. Some of them were performance markers, but many of them were blood markers. We stipulated that we had to get very fresh blood. The blood had to be from the vein of the participant back into the lab in a maximum of 1 hour, so that we could analyze cells that were still pretty much as they would have been in the person's body. I'm not going to go into the details. This is a complicated table, and the paper was just published in Mechanisms of Aging and Development. But we found that although we could identify quite a number of markers, we didn't come up with yet the kind of the gold standard we want of something which can really tell you how an individual is progressing. We haven't yet analysed the longitudinal data, and we're hoping that something will come from that. But what we did find when we analysed the baseline data was that there were some really unexpected negatives. Inflammation. We believe inflammation to be important. We believe TUC. We believe others. But actually the associations of the inflammatory markers that we had with the various age-related outcomes did not really show up as being meaningfully significant. short telomeres. Interestingly, within very old people, and this had been shown before in a collaboration with the Leiden 85 plus study, when you get to be 85 and above, telomere length does not associate with risk of morbidity and risk of mortality. This is partly due to the changing clonal makeup of the blood cell population. There is something that's known as the immune risk profile associated with low survival in Swedish longitudinal studies of older people. We did not find the immune risk profile to associate with health status in the Newcastle 85 plus study. So it remains something that we would really like to know, but we do not yet know, which is how to be able to get a grip on the changes that are occurring. Genetics. Now this is something that we really feel we ought to know. We live in the era of the genome. So of course we should know what genes influence human longevity. We know from twin studies that genes account for 25, maybe 30% of the variance in human lifespan. So surely it should be easy enough to get to grips with what those genes may be. Now, back in 1993, I was involved in a collaboration with François Schachter and Daniel Cohen at CEF, and we wrote a paper in Human Genetics that kind of set out strategies that we might adopt in order to be able to dissect the genetics of human longevity. And the year after that, in Nature Genetics, the CEF group published an association of the ApoE polymorphisms with in a case control study, centenarians and non-centenarian controls. And we know that if you look across the life course, that as you go to higher ages, the frequencies of the different ApoE alleles changes in ways that are kind of broadly consistent with that result. And also, there are quite a large number of studies now that are performing genome-wide association studies of long-lived individuals, centenarians, and 90-plus SIB pairs family groupings. Now, when we look for genes associated with longevity, if there are genes that are pro-longevity, then we would expect to find them overrepresented in centenarians, and that's the case example of that is ApoE. We would also expect to find in people at very old ages that where there are known genetic risk factors for a spectrum of age-associated diseases, heart disease, diabetes, and so on, we would expect to find that those who survive to very old ages should have fewer of the risk alleles. Broadly, what we would expect is this. If you imagine that you've got genes that are associated with longevity, genes that are associated with disease risk, and genes that are normal, as it were, you would see that as you went to higher ages, the proportion that are associated with longevity would increase. The total number, of course, would decline because some mortality occurs. And the ones that were associated with disease risk should have sort of dwindled out of the population, and the normal ones will, of course, have reduced. So this is the kind of a priori expectation. What then do we make of results like these that came from the Leiden group that has done some of the best studies on genetics of human longevity, published in PNAS last year? Genome-wide association studies failed. Those risk alleles that had been previously reported in GWAS were present at equal frequency in the population. So nonagenarian siblings from long-lived families and singletons older than 85 years from the general population carried the same number of disease risk alleles. Longevity in this study, they reported, was not compromised by the cumulative effect of the risk alleles for common disease. Tuck Finch and I, when Tuck visited Newcastle last year, decided that we needed to articulate some of our concerns about where things were heading in terms of the expectations of genetic studies. And we published a paper just a few weeks ago, Trends in Genetics, with the rather sort of gloomy, perhaps, but we think realistic title, Speed Bumps Ahead for the Genetics of Later Life Diseases. And we think that within the field, and we were astonished to discover that our co-author, Heather Cordell, who's one of the UK's leading figures in studying the genetics of complex common disorders, was actually at the start of the interaction, was simply not aware of what we know about the genetics of longevity. And most of the current research in this simply overlooks caveats that are indicated by the science of longevity. And the science of longevity tells us that we should expect a highly polygenic mix of largely unselected genetic variation underpinning this array of complex traits. And we should expect that a substantial fraction of the phenotypic variance in aging arises from intrinsic chance with no obvious genetic or environmental source. So we are going to have to become a whole lot cleverer if we want to be able to attribute the 25-30% heritability of lifespan to specific SNPs than just simply doing these highly technically sophisticated, but ultimately, I hesitate to use the term, but rather brainless, sort of screens. It's not that people don't have brains, it's just that they've not got them connected to the right issues. Something that, again, causes me a certain amount of trouble is that we are seeing within the field the emergence of quite a number of genetic models of accelerated aging. Now, we know that lifespan is regulated by multiple genes that code for different maintenance and repair functions. Lots of different DNA repair pathways, lots of different antioxidant pathways, you name it. So what you can do is take one of those processes that you believe to be important and break it. You introduce a mutation in a pathway that you think should accelerate the accumulation of lesions that you hypothesize are associated with aging. And this is fine, it works pretty well. Jan Hoymaker's group has done some beautiful work in transgenic mice deficient in individual genes for DNA repair and transcription. And what they see is premature aging. And another example is the beautiful work that came out of Niels Goran Larsen's group, and Sandra Trifunovic led this work. She now has her own lab, developing the Pol gamma mutator mouse, generating mutations in mitochondria. But what I think we're not taking sufficient account of in these models is that, first of all, we have to be very careful that the accumulated lesions are similar in nature to those that occur during normal aging. And secondly, those lesions, if they're driven by breaking one particular part of the machinery, we may be missing all the synergistic interactions that we saw from the cell senescence work are important in what happens in the animals. And there is a slightly disturbing trend to make a transgenic mouse model in which you break something and show that it ages faster. And then you do an experiment to show that you fix what you've broken, and hey presto, it lives as long as it did to begin with. And that gets touted as a potential therapeutic intervention for aging. And I think, again, I don't want to belittle the beautiful science that's done in this area, but I just think we need to be a little bit cautious, because I don't think we really know what we sometimes think we really know. So, outstanding challenges. There's a lot. How multiple mechanisms interact to drive aging, systems biology, plus. The multi-level causal connections between intrinsic aging and pathogenesis. We really need to understand how these things work. What are the perturbations to molecular and cellular homeostasis that underpin these connections? Where the pathways in different age-related diseases overlap. Because again, there's probably huge information to be developed by bringing people together who study different diseases. We're trying to do this within our Biomedical Research Centre for Aging in Newcastle, where we've got work going on in parallel on 7 different age-related disease areas, and we're trying to understand the commonality. What determines the tissue and organ specificity of the mechanisms linking aging to disease? I think this is one of the huge unsolved areas in the biology of aging and of disease. Why do certain lesions produce pathology in just one organ or one subset of organs? How to establish the role and limitations of genetics, how to get the biomarkers I mentioned. Ultimately, we want therapies. So which therapies might target the fundamental processes, which might deliver efficacy, and how exactly lifestyle and nutritional factors affect these fundamental processes. We have the epidemiological associations. You know, we know that the Mediterranean diet, huge study on 33,000 people in Europe, showed that if you score people's adherence to Mediterranean diet on a 10-point scale, then if you move 2 points on that 10-point scale towards a more Mediterranean diet, you see an overall reduction of 8% in mortality across the life course. Unquestionably, it's true, but how does it work? We don't know. So, in the abstract, I referred to this guy, a very important figure in science, Nobel Prize winner, one of the first people to sort of bring in the modern era of study of aging. But he also had a habit of being rather schoolmasterly and telling people off. He did this in a book that he wrote later on in his life called Advice to a Young Scientist. And I hope none of us will have the temerity when we become old to pen a book offering advice to a young scientist, particularly because in this book he used the book to get back at people he had disliked earlier in his life. But Medawar wrote in 1955, "Nothing is clearer proof of the immaturity of gerontological science than the tentative and probationary character of its system of definitions and measurements." Well, it probably was true in 1955. Arguably, there are things that we maybe need to measure better now in 2011 and beyond. 10 years ago, I was given the extraordinary honour in the UK of being asked to deliver the Reith Lectures. Now, the Reith Lectures, in the US, you may not know what they are. The Reith Lectures are they really are the Nobel Prize of broadcasting in the UK. They're a series of lectures given annually to commemorate the first Director General of the BBC, Lord Reith. They're broadcast in the entire English-speaking world except the US. They're done on radio rather than television because in Reith's day there was only radio. And I was absolutely delighted, honoured and amazed to be asked to deliver the lectures on the topic of ageing. Now, although you didn't get the chance to hear them here in the US, thanks to the wonderful technology at the BBC, you can actually hear them. You can go to the BBC website, the link is at the bottom, and they're recorded. You can play them. You can even get the question and answer that comes at the end of them. But in the last of these lectures, New Directions, I've reproduced just some of the text. You can also get all the text from it. And I think there is a really important point that we within the field need to recognise. Now, we do sort of recognise it, but I was inspired by Bob Butler's term, the longevity revolution. So I use this as a kind of reference point. But a revolution has a turning point, a time when the original impetus for change has run its course. And this is a risky time. Now, the increase in life expectancy that we've experienced in the human population comes from being unwilling to die. We're scared of death. We've run away from death. We've been spectacularly successful in running away from death. We have increased life expectancy by 2.5 years, 2 to 2.5 years, for every decade of the last 200 years. Life expectancy here in the US and the UK is going to increase today by 5 hours because we are so very good at running away from death. But you can't keep running away forever. You have to ask yourself, where do I want to run to? And this, I think, is a major challenge within our field. So I'm going to be slightly cheeky, and I'm going to start to address, sort of what I believe to be a great lack. What do we really want? We say, glibly, that we want to maximize the health span within the existing lifespan. Okay, but what does this really mean? If you could do it, people used to talk about rectangularizing the survival curve or becoming like the old Oliver Wendell Holmes one-horse shay, remaining in perfect condition until one day you drop dead. Well, if I was in perfect condition, the last thing I would want to do the next day is drop dead. And I think there is a central problem in this conception. We want to extend health span and lifespan, and we point towards calorie restriction as a way that does this in laboratory animals, and there's a whole lot of discussion, we've seen it here, about CR mimetics trying to do this. But the problem is, okay, you might push the bad stuff away, but you've still got to deal with it. It's still going to come along. Calorie-restricted animals die just the same. They die with the same spectrum of disease and the same impairment of quality of life, and that will be true for us if it's possible, which I'm not sure it is. And then you get people who want to eliminate aging altogether, and to be quite honest, I have very little patience with this particular school because I think it's an unrealistic distraction. And even if we one day were going to be able to get there, we would need to navigate the above two stages first. I have written about a world in which it's possible to eliminate aging altogether. If you read my book, Time of Our Lives, Science of Human Aging, there is a short story at the end of that, Miranda's Tale, which describes a world in which this has been possible. It also, if you read it carefully, it gives a pretty realistic, in my mind, sense of the timescale before we will reach that point and of some of the pitfalls that lie between. I've sold the film rights, so one day, if you don't want to read the book, you might be able to see the film, but the filmmaker that I've sold the rights to seems to be rather slow about getting on with the job. So don't count on it. And I didn't make a lot of money by selling the rights, but I do get a big share of the profit. So I'm coming to the end, and I didn't pay attention to when I started, so I apologize if I've gone on longer than you were bargaining for. I have a number of heroes, and one of them is this guy, Lord Rayleigh, 1904 Nobel Prize in Physics. And he said, one should neither seek nor avoid complexity in finding the appropriate solution to the problem at hand. And I think that is an absolutely pertinent principle for us in aging. And now, very much tongue-in-cheek, I'm going to offer an update on the Medowa view. And that is that nothing is clearer proof of the immaturity of gerontological science than the tentative and probationary nature of our current ability to define our true goals and of our capacity to engage with complexity to the necessary extent. So I'm being slightly arrogant in paraphrasing Medowa's words here, but the second part of it I really believe. I just think we owe it to humanity to take this agenda further forward. So I think I'm done, and I'd like, just in closing, to thank some people for inspiration and support. As they say at the Oscars, of course it's a family, but actually in my case, my father was an outstanding individual. He was South African, born South African, and after World War II he committed himself wholly to fighting the threat of the nationalist takeover of the South African government and the introduction of apartheid. And he was pretty fearless and pretty outspoken, and he became Oxford University's first professor of race relations. And I'm really grateful to him for providing a model of what one needs to do to make change. My mother, during the last years of her life, spoke to me a lot about growing very old, about being frail, about the meaning of life, about the process of dying. And that again I found enormously valuable. My kids who are far more impressive at the age of 27 than I was at that age, have taught me a lot about being a person in today's world, and my wife Louise, for everything. But there are a number of individuals who have really inspired me. David Kendall was an extraordinary statistician who taught me at Cambridge and really excited me about stochastic processes in biology. John Kendrew's wonderful book, The Thread of Life, to which I was referred by Robin Holliday, fired my enthusiasm. Leslie Orgel, John Cairns, John Maynard Smith, wonderful scientists, each of them truly inspirational, and many, many people in this room. But I want to close with an acknowledgement to my real scientific hero, and this is someone that I call the unknown scientist. And the unknown scientists, there are loads of them around. These are the people who may never hit the scientific headlines, but they are the people who make science happen. They're the people who embody the real principles of science, which I've listed here. Humility in the face of the unknown, dedication to truth, the resilience to keep going through the ups and downs, and we all know that there are many, many more downs than ups, and openness to new data and ideas. So thank you very much for being patient and listening to me.